Hello, and welcome to this week's seminar of the Living Earth Collaborative and Washington University's Evolution, Ecology, and Population Biology group. It is my honor to introduce Dr. Adam Smith as today's speaker. Dr. Smith is an associate scientist with the Missouri Botanical Gardens Center for Conservation and Sustainable Development, where he is the PI of the garden's Global Change and Conservation Lab. Dr. Smith received his master's from the University of North Carolina at Chapel Hill, his PhD from the University of California, Berkeley, and thereafter completed postdocs at both UC Berkeley and the Missouri Botanical Garden. Dr. Smith's research addresses the impacts of global change on individual species and their communities, and the means by which human intervention can facilitate adaptive responses. A dominant theme of his work is the development and application of models that predict biogeographic responses of species to climate change, habitat loss, and other modes of environmental change. As a testament to his prolific research career, Dr. Smith is published in myriad journals, including Nature and Nature Climate Change, Trends in Ecology Evolution, and Global Change Biology, among others. Dr. Smith is also an ongoing contributor to reports for the IUCN Species Survival Commission. Alongside this impressive research career, Dr. Smith is dedicating to teaching and mentoring. He has provided mentorship for 8 PhD students, 6 of whom are current or former WashU students, 4 postdocs, and multiple undergraduates and postbaccalaureates. He has also taught global change conservation at the University of Missouri St. Louis, spent 3 years teaching English in Japan, and was awarded Professor of the Year at McPherson College. As one of Dr. Smith's postdocs myself, I can attest to his extraordinary capacity as a mentor. Notably, Dr. Smith is also well integrated into our Living Earth Collaborative community. He is both an LEC Fellow and a recipient of an LEC Seed Grant. Now, before we begin, I'd like to remind everyone in the audience to please leave your questions and comments for Dr. Smith in the YouTube live chat, and we will relay them to him after his seminar. And with that, thank you, Dr. Smith, for joining us today. I will hand the floor over to you.
Thank you very much, Matt. That was a very kind introduction. And I'm very happy to talk with all of you today. I'm very honored. Let me get my interface set up here. There we go. So today I'd like to talk with you about a dearth in our knowledge about the fundamental requirements of species, and that is known as the Hutchinsonian shortfall. But before I begin, I want to advance to slide 2. There we go. Before I begin, I want to say thank you to a very special person, and that is Alan Graham. Alan unfortunately passed away very suddenly last year. Without Alan Graham's beneficence, my position and the Global Change and Conservation Lab at the Missouri Botanical Garden would not have been possible. So I want to give a great debt of gratitude toward Alan. And Alan's work not only sponsors my research, but also research of plenty number of people, including Matt Austin. But of them, we have Lauren Jenkins, who's interested in reconstructing species biogeographic histories across the last 21,000 years and examining their responses to climate change. We have Matt Austin, who's also co-sponsored by Ken Olson, or co-advised by Ken Olson, and Kelsey Bartlett, whom both are interested in examining how species' reproductive capacities have changed across the last century in response to climate change. We have Kelly Erickson, who's tackling what I would say one of the hardest problems in ecology, and that is understanding species responses to the environment when we know almost nothing about species. We have James Lucas, whose major advisor is Robbie Hart. James is interested in the future of paper-making plants in Nepal and the paper-making industry and how that might change with climate. And we have Ethan Abercrombie, who last year did a rotation in my lab and is interested in seeing how communities of mammals have tracked or not tracked climate across the last 100 years in the Sierra Nevada of California. Now, I present to you this short menu of the research we've done, not just to say thank you to Alan, but also to emphasize the point that each and every one of these lines of inquiry really depends upon understanding species' fundamental responses to the environment. And indeed, there's probably not a single line of inquiry in ecology that doesn't fundamentally depend upon that. Unfortunately, we have a profound lack of knowledge about species' actual responses to the environment, and this is called the Hutchinsonian shortfall. But it's in good company. There's a lot of other lacks of knowledge about ecology. Starting on the right, we have the Linnaean shortfall. This is lack of knowledge about simply what species are out there. For most species on Earth, we don't have a description or a name. We have a Darwinian shortfall, which is lack of knowledge about species and evolutionary relationships. Eltonian shortfall, lack of knowledge about species interactions. And then the Wallacean shortfall usually refers to lack of knowledge about past and current distributions of species, but I'm going to split that up. So I'm just going to assign the Wallacean shortfall to lack of knowledge about current distributions. And then in honor of Margaret Davis, who did a lot in this realm, assign the lack of knowledge about species past distributions to her. And then finally, we have the Hutchinsonian shortfall, which is the topic of what I'm going to speak to you about today, and that is lack of knowledge about species responses to the environment, i.e. lack of knowledge about niches. So simplistically stated, we might think about a niche that looks like this. And I should say that here on out, there are many different ways to define the niche, probably just as many different ways to say the actual word, but I was speaking mainly about the climatic niche, that is the thermal regimes, the moisture regimes, what have you, that allow populations to at least persist, if not grow, in environmental and geographic space. So a niche, in a cartoon fashion might look something like this. In this green area, we have positive growth rates. In this red area, we don't have data on that area. And this is what I'm going to be talking about, whether or not we can infer whether species can persist in these locations, even though prima facie, we don't really have information on those locations. What causes this? What causes the lack of information about species' fundamental environmental tolerances? Well, a lot of it, unfortunately, has to do with these other shortfalls. For example, if we don't know if the species exists or not, it's almost a moot point to talk about what its niche might be. If we don't have information on its interactions and its former and current distributions, that makes it much harder to infer what the species might actually prefer. So the Hutchinsonian question, and Hutchinson had a lot of questions, but the Hutchinsonian question I want to address today is, what are species' fundamental environmental tolerances? Now, a rhetorical question in response to that might be, well, why not just go out and measure them, do laboratory experiments or field experiments? And indeed, that is really kind of the gold standard in terms of ascertaining what kind of conditions are conducive to positive population growth. That said, for the vast majority of species on Earth, like it or not, it's just not going to be possible to measure what their actual fundamental niches are. To give you an example of how difficult this is, even for the species that we are studying, let me illustrate the point by pointing to a recent review we conducted of common garden experiments. Common garden experiments are usually used to detect local adaptation. So it's one component of kind of deciphering the Hutchinsonian shortfall. So in this case, we did our literature review of all the studies that we could find that did common garden studies experiments on plants. And what we found was that the modal study used a single species, not more than that. It focused on trees growing in monoculture, so not relating to any other species in a meaningful fashion. They sourced material, meaning seeds or seedlings or what have you, from 3 sites and only grew them at 1 site, which is almost inadequate really for understanding species responses to the environment. It lasted only 3 years, and they usually did not conduct any additional manipulations like rainout shelters or something like that. So this just goes to show you, for even the species that we are studying, our knowledge is very, very limited. And as a result, for the vast majority of species on Earth, we really can't rely on this kind of data, the gold standard kind of data. Rather, what we do have for quite a few species on Earth is occurrence data. And by occurrence data, I mean herbarium data, museum data, and community science data, or any kind of other kind of observations. So this kind of data doesn't cover every single species on Earth, but it is fairly voluminous. Per force, we have in the Global Biodiversity Information Facility the database of all the records in across all the museums, or at least it's tended to be on Earth. We have over 2 billion records of occurrence of species. This is a map of just those occurrences. It is highly biased. You can see that basically very rich countries have a preponderance of occurrence data. It's not that we have more species or individuals there. It's just that we have more people and resources to go out and find these locations. So there's that issue, but this is the fundamental data that we have for a vast majority of species. In my talk, though, I want to extend the idea of occurrence beyond just this kind of data, but also talk about what I would call extended occurrence data. And that would include pollen data, fossil pollen deposits, and of course, DNA data. As long as it can, any of the kind of data can be mapped, it is occurrence data, at least in my book. So the two questions I would like to address today are this. To what degree are species climatic niches truncated by geography? I'll explain what I mean by that. And secondly, how can we leverage this extended kind of data, pollen and DNA and so forth, to overcome the Hutchinsonian shortfall? So let's address niche truncation first. And I'm going to explain this by kind of walking through my process by which I was confronted by this issue. It was always something in the back of my mind, but it really came to the fore a couple of years ago when I stumbled across several tomes, each of them several hundred pages in length, all published by the US Forest Service by Robert Thompson and others. And each of these tomes at the beginning has maybe half a page or a page of text only explaining generally what they are. So I'm still honestly don't exactly know why they were produced. It's somewhat of a mystery to me. But each of these tomes has page after page after page that looks something like this, one per species or one for a group of species. So let me walk you through these through one page, and I'll show you exactly what's really capturing my interest. So this is the page that focused on green ash or Fraxinus pennsylvanica. We have, of course, a species range map. These range maps, by the way, were produced by Egbert Little in the 1970s, and they were on government servers up until 2016 when the great climate data purge of government servers occurred. This obviously isn't climate data, but somebody luckily scraped it before it was erased from posterity. Anyway, this is the species range map. And this is the map of the species in geographic space. I'm going to be, excuse me, I'm going to be going back and forth quite a lot between geographic space and I'll call it environmental space or niche space. So niche space is what is mapped in the rest of each of these pages. And this is just a short section of these. So up here at the top, we have the species occurrences in terms of temperature. And down here at the bottom, we have the species occurrences in terms of precipitation. So let me focus on the top set here. We have three little boxes. The first pertains to annual temperature. The second to January temperature. You can think about that as winter temperature, the coldest temperature of the year usually. And the third to July temperature, usually the warmest temperature of the year. And in each of these boxes, we have a set of lines. They're actually a bunch of dots all kind of smushed together, looks like lines. The top line or set of dots represents the temperatures where the species currently occupies. And the bottom set in each of these boxes represents the temperatures where the species is absent all across North America. So we can look at where it's distributed in the annual temperature, January temperature, and July temperature. In other words, this is our best representation of the species thermal niche. Now, we can use this information to, in a sense, forecast species' potential responses to past or future climate change. For example, we can ask, what is the breadth of temperatures that this species, green ash, can tolerate? Well, we just need to look at these two graphs. So this is the species distribution across January temperatures or cold temperatures, and the coldest temperature that it can be found at is at about negative 25 degrees Celsius. The hottest temperature it can be found at is at 30 degrees Celsius. And we can use this very simple, I would call it a niche model, to forecast what might happen to a particular location. Let's assume that we are standing at a location that's currently 30 degrees in Celsius. Now, it's at the very tip top of the species thermal range. And we can ask ourselves what's going to happen if this advances to, say, 32 degrees. Well, we need just look over here at this portion. We say, well, when temperatures get up to 32 degrees in July, the species doesn't exist there. And so lacking any other kind of information, we would conclude that the species would in turn suffer. And we can say that because we have absence data, in a sense, above temperatures of 30 degrees Celsius. And conversely, if we were looking at colder temperatures, we have absence data at temperatures lower than negative 25 degrees Celsius. In other words, the species niche is, in a sense, encompassed by the rest of the available environment. And this seems to be true page after page, species after species, when you look at these univariate responses to climate. The picture changes, though, a lot when you start looking in more than just one dimension. These are the exact same variables, but now we're plotting one against the other. So here we have July temperature, summer temperature, and here we have January temperature or winter temperature. This gray area represents the locations across North America where the species is completely absent. These dark dots represent the places where the species is present. And species after species, what really sticks out to me in many cases is this area, this gulf. And I'm going to call that area an area of potential niche truncation. In other words, it really seems like that if there were climates that appeared over here, it's very possible that species could exist there. But because those climates don't currently exist on the landscape, we don't have any information about whether the species could tolerate those environments. Again, you could do some sort of manipulative field experiment or laboratory experiment to find that out, but we're not going to be able to do that for the majority of species on Earth. So what kinds of, or what causes niche truncation in the sense that I'm talking about it? I'm going to speak about two different kinds of niche truncation. The first of these I dubbed the range edge is synonymous with the niche edge kind of truncation, or AKA the problem of Florida. Let me illustrate that with Florida. This is a map of Florida, or really actually a picture of Florida. And of course, as you know, as you go north, things get cooler. As you go south, things get warmer. Let's take, for example, the distribution of this species, or really it's a variety. This is the Carolina petunia. I'm going to make the strong assumption or weak assumption, however you see it, that species distributions are currently limited only by climate. That is completely false in many cases. We can think of many different examples where that's not true. And yet, like I said, for the vast majority of species on Earth, we don't have other information on what might be limiting species distributions. When those ranges are limited by non-climatic effects, then everything I'm going to show you from here on out is actually going to be exacerbated. In other words, what I'm going to show you is the minimal amount of niche truncation that occurs. And when you add in, say, lack of species on which our focal species might depend on, or add in competitors or disturbances or things like that, that might also truncate the niche, the situation gets far, far more exacerbated. So let's go back to this species and let's presume again that its range is entirely distributed or entirely determined by temperatures. So we can ask ourselves, what's limiting the species range? Well, as you go north, you run out of species and things get colder. So we would conclude that the species distribution on the north side of the range is limited by cooler temperatures. What about warm temperatures? Well, as you go south, you run out of Florida, and therefore we can't tell necessarily whether this species really meets the edge of its thermal niche at the edge of Florida, or whether it could persist in even warmer temperatures. Now, of course, you could conduct a manipulative field experiment, a transplant experiment, but I hope you can see why ecologically that would probably be irresponsible to transfer it to an island south of here in the Caribbean, and depending on where you'd like to take it, perhaps even politically difficult. So this is an example of what I call the range edge being synonymous with the niche edge. On the other hand, we have causes of geographic niche truncation that I call extreme interiors. And I'll tell you how I stumbled across this. I was working with Christy Edwards and others at the Missouri Botanical Garden to understand the phylogeographic history of this species. It's an endangered species, Delphinium exaltatum. And I constructed a niche model for this species, and it looks something like this, at least its response to water availability. So on the x-axis, we have amount of water that's available to the species, and on the y-axis, we have the environmental suitability. And as you can see, as things get wetter and wetter, the species gets happier and happier, and then we run out of water, so to speak, or rather, we run out of conditions that are even wetter than this. Where does this occur? Where does this extreme amount of wetness occur? It occurs right here in the southern Appalachians, actually tend to be the wettest part of the southeastern US. That means that if I have to extrapolate my model into even wetter conditions, I don't really know what the behavior is. In other words, the species might get happier and happier, or it might peak in happiness, so to speak, and then become sad because things are getting too wet. In practice, usually things don't get too wet per se. Wetness or rain precipitation corresponds with too much cloudiness. But regardless of what is actually causing this, I don't know the trajectory of the species in wetter conditions. And this really illustrates the fundamental difficulty of niche truncation. We can only see up, in a sense, up to the edge of the precipice, and beyond that, we have to extrapolate into regions for which we have no data. Now, there are other causes of niche truncation I've already mentioned. We can have biotic interactions or lack of biotic interactions. We can have dispersal limitation. We can have human interference. People could remove species from landscapes where they would otherwise be occupying. I'm not going to mention those any further because I've already discussed them, but like I said, when we consider these aspects, then you can see that everything I'm going to talk about is actually exacerbated. So let me walk you through the process by which I explored the degree to which species realized niches are truncated by geography. And I'm going to do that with this species. This is Senegalia greggii. It used to be called Acacia greggii, so it's an acacia species. The common name is the wait a minute bush, and that's because it has these really sharp talons. If you're walking by them, you often get caught and you have to say, wait a minute, while you disentangle. And it has a name befitting basically the way I'm going to use it as a demonstration. So this is the range of the species. You can see it's distributed about half of the extent in southern US and the other half in northern Mexico. I took from the species range all the environments associated where it's located. And to do that, I'm not going to walk you through the nitty-gritty details, but basically I use 17 bioclimatic variables that seem relevant to the distribution of the species. And then I ran them through a principal components analysis, and then I mapped them onto this. This is our environmental space. So this region here represents the species realized niche. In other words, these are the climates that the species currently occupies. Then I was interested in where the realized niche sits vis-a-vis or in relation to available climate space. So to examine that, I took climates all from across North America and did the exact same thing, ran them through the principal components analysis, the same one, and then mapped them onto this environmental space. And as you can see, the species niche sits in a sense on the edge of that area that's accessible to the species. And given this, we can start to ask ourselves, to what degree is climate favorable or unfavorable to the species? So obviously, if a place has a climate that's in here, it's favorable to the species. But if the climate's out here, prima facie, given the evidence we have, given occurrence data, tabula rasa, whatever Latin phrase you want to use, vidi vidi vici, you'd conclude that the species does not exist and cannot exist in these environments. Now, in reality, maybe it just hasn't been able to disperse there, or there's a competitor that keeps it out. But again, like I said, given the data we have, this is just what we'd have to conclude. You'll notice the species exist on the edge of this climate space. Very particularly, we can ask ourselves, would the species like these climates out here? Would it persist if those climates were actually to exist? And the answer is we don't know, but we can at least identify, in a sense, where the limits of our knowledge end and our ignorance begins. And that's the point of niche truncation. So if I could zoom in on that, it's literally right here. The species literally exist on the precipice of the currently available climate. So to quantify this amount of niche truncation, I calculated the perimeter of the species realized niche and then the proportion of that perimeter that faces climates that do not exist currently on the landscape. And for this particular species, about 12.5% of the perimeter faces those climates that don't currently exist. So in other words, 12.5% of the perimeter is truncated. And then I repeated this analysis for about 650 North American trees. And I have a note here that says these are bars, one per species, ordered from least to most truncated. To remind myself to tell you that these are bars, one per species, ordered from least to most truncated. And let me show you exactly what I mean. These are bars, and the height of each bar represents the proportion of each species niche perimeter that is truncated. It faces climates that don't currently exist. And that species we were just looking at is actually that bar right here. So this is the pattern of truncation across 650 North American trees. And we can calculate our summary statistics. On the whole, on average, it doesn't seem so bad. Only about, on average, 11% of a species niche is truncated. But it ranges up to, depending on the species, up to 2/3 of the species niche being truncated by current available environments. About 2/3 of species had non-negligible truncation, And of those, about 1 in 10 had what I would call severe truncation. That is, up to a third or more of their niche is facing climates that don't currently exist on the landscape. In other words, about a third of the species have or pose, in a sense, a profound lack of knowledge about the responses to potential future climates or potential past climates. Let me walk you through two of the species to just show you what this looks like. This is the most truncated species niche-wise. So this is Sitka spruce. It's located along the Pacific Northwest in the temperate rainforest. And even though this isn't, in a sense, any peninsula, it does have some islands, but they're not really kind of large islands. Climatically, it sits on a peninsula. So in geographic space, it's more or less a line along the coast, but climatically, it actually really sticks out. It's very different from any other climate available in North America. And as a result, the species niche is severely truncated. In contrast, this is balsam fir. This species also has a range that abuts the coast, but climatically, it's still its niche, realized niche is still totally encompassed by available environments. So we can use that information with more confidence than the previous species to predict its responses to past or future climate. Now, this all said, we need to ask whether our ignorance in this area is really relevant. In other words, if we're trying to construct a model of species niche and say we're trying to project that model either to the past or the future, if past or future climates don't appear out here, then our ignorance is kind of a moot point. It doesn't really matter that we don't know those responses because the species is not going to experience those climates in that area. So to ascertain whether whether niche truncation is relevant or not, looking forward, I looked at climates of the future and then I mapped them again to this niche space, and I kind of accumulated these through time. So this rose-colored area here represents climates from 2025, 2055, and 2085 under this particular climate scenario. And you can see they expand from available niche space, meaning we'll have novel climates in the future. And some of these novel climates appear right in the area of niche truncation right here. Now, let's focus on this area over here. Technically speaking, if I were to construct a model of the species niche and to project it into the future into these climates, wherever they are located in North America, I would be extrapolating that model. However, personally, I would feel generally confident in my predictions if that model said the species can't exist there, because it would be really strange if in reality the species could persist in these environments and then suddenly pop up and exist in these environments. That just doesn't seem very possible. In contrast, I would have a severe lack of confidence in the model when I'm predicting to areas that have environments indicative of this area, because I don't have information about how the species could respond to those environments. So that's risky extrapolation. So I repeated this procedure for all 650 species. And let me show you the results. Now, this is the exact same slide I just showed you earlier, but I've color-coded everything gray so you can do a kind of a visual comparison. Now I'm going to add in, overlay on this, the degree to which each species truncated, or the portion of the niche that's truncated, is actually facing future climates that do not currently exist on the landscape. And that looks like this. So each of those reddish portions of the bars represents, in a sense, risky situations where future climates are going to appear that the species doesn't currently experience, but we don't really have information on how the species respond to those future climates. And again, we can calculate our summary statistics. And honestly, they look very, very similar to just kind of normal niche truncation, regardless of whether you consider future climates or not. So on average, about 10% or so of the species niche faces future climates that don't currently exist. On average, about 2/3 of species are truncated in their niche. And on average, about 1 in 10 species faces severe niche truncation vis-a-vis future climates. We can also do this for past climates. So again, I did the exact same thing, but now I'm running back. And in this case, I used climates from every 1,000 years, going back to 21,000 years ago. 21,000 years ago is kind of like a magic starting point, because that's the point at which the glaciers from the last glaciation were at their maximum extent, and everything since then has been kind of shifting in response to their retreat. So in this case, for this species, you can see that niche truncation, even though it still does occur, in relation to past climates, it doesn't really matter so much because the species truncated portion of the niche doesn't face those past climates. So any kind of model is probably going to be more reliable if we backcast it to the past than if we cast it to the future. So across all of our species, these are the responses, or this is the degree to which each of these niches are truncated vis-a-vis past climates. And again, the summary statistics are very, very similar to what we've already seen. About 1 in 10 species faces severe niche truncation. Now, given all of this, I became interested in where in geographic space this kind of niche truncation is occurring, regardless of whether you're facing either the past climates or, in a sense, the future climates. So where are these environments associated with truncation? So to do that, what I did was I located the points of truncation and then I mapped them to the map of North America. So basically I'm taking the environment right here along the species niche as it faces outward, so to speak, and putting it onto the map. And here is that map. These pixels here are color coded by the number of times that location represents an environment that sits literally on the edge of the species observable niche. It goes up to about 10 species. And you can see that there are kind of hot spots of locations where a species niches are truncated. Let me focus on one of those. We've already talked about it. This is the Pacific Northwest. You can see there are quite a few species and quite a few locations that, in a sense, face outward from available niche space into, in this case, I was looking at past climates, but the map for future climates looks very, very similar, so I'm not going to show it. You can see there's quite a few places where truncation occurs in the Pacific Northwest. And again, this is because climatically speaking, the Pacific Northwest sits on an island, even though geographically, or sits on a peninsula, even though geographically it's not actually part of a peninsula. So in summary for this section, to answer the Hutchinsonian question, to what degree are climatic niches truncated by a geography? The answer is, on average, maybe you'd say maybe not a lot, and yet for a non-negligible number of species, we do have what I would call severe niche truncation. About 1 in 10 species faces climates that don't currently exist in the landscape, but either have in the past or will in the future. And quite often, this truncation either occurs in extreme environments, such as the southern Appalachians, which are extremely wet, or it occurs along coastlines. This is important because it poses a fundamental challenge to our ability to predict past and future species responses to climate change. And it's something I run into quite a lot. I don't have a gold key to solve this problem, but I want to walk you through several different initiatives that we've been trying to tackle this issue in our lab. I will mention before I go any further that it's, even though we have other shortfalls which are feeding in a sense to the Hutchinsonian shortfall, it is possible sometimes that we can take knowledge indicative of these other shortfalls and use it to shore in the Hutchinsonian shortfall. So for example, one of the initiatives in our lab that we've been working on, and this is work begun by David Henderson, WashU student who did a rotation in our lab a couple of years ago, is using herbarium and museum specimens that are commonly discarded before biogeographic analyses. So quite often herbarium and museum specimens were collected by people interested in taxonomic science. And especially if they're collected, say, more historically, the ability to geolocate, that is to locate those particular specimens to a particular point in space, can be very, very challenging, especially older specimens where people generally thought, well, it's okay just if I record the county, say, or something like that. That makes it very difficult, if you only know the county in which a species was located, to match it to environment in that county, because any particular county can experience a lot of variety in the climates that it covers. And as a result, general guidelines for using this kind of data stipulate that this kind of data should be thrown out. That is, records that can be only geolocated to the county level or something like that. Unfortunately, that can represent a very large proportion of records, depending on the analysis. Literally, not kidding, that can mean throwing out up to 97% or more of the records out there. And that represents tremendous loss of human effort and human knowledge about the distributions of species in geographic space, and by in turn, distributions of species in environmental space. Here's just one example. What I'm showing you here is a map of this particular species. This is Asclepius amplexicalis, a milkweed. The occurrences that can be geolocated with confidence are these small green dots. And to help you visualize them, I've drawn this minimum convex polygon around them. If we were to restrict our analysis only to these precisely geolocated records, you can, and then say examine the species' environmental tolerances, you can see that they do not extend as far south as the species actually exists. And therefore, we'd conclude that, for example, it can't tolerate hotter temperatures that are indicative of these locations down here, nor can it tolerate colder temperatures that are indicative of these locations down here. So you can see how that would artificially truncate the niche through sampling. In contrast, if we don't throw out those records that people normally do, then we can probably better grasp the species' true extent of its distribution in environmental and in geographic space. So this is, in a nutshell, what we're doing to help tackle this issue. Basically, it's using data that people normally think of as useless because it actually has great use. Another initiative that we've been tackling is our initiative method we've been using to tackle the Hutchinsonian niche is by leveraging phylogeographic relationships between species. So for example, maybe you have a species with a niche that is severely truncated by geography or something like that. If there are closely related species whose niches might be more filled, so to speak, then I can use that phylogeographic information or phylogenetic information to help infer the real niche of the species that I'm interested in. Another initiative that I've been engaged in, and this is something I'm really excited about, is using species past distributions to help fill in our lack of knowledge about species' fundamental environmental tolerances. And this is a great collaboration. It's really kind of set the bar for me in terms of collaboration that I'm engaging with all these friends/colleagues shown here at the bottom. And this is work funded by the National Science Foundation. So let me walk you through how we actually use species, I would call it past distributions and that kind of data. It's basically extended occurrence data. So it's DNA and pollen and things like that to infer what species' true environmental tolerances are. And I'll do this through a cartoon example by referring you to this, this plot I showed you a little bit earlier. And like I said, we don't really know in this case whether the species likes even wetter conditions or even drier conditions or what have you. Pardon me a sec. So this possibility represents alternative realities. And I'm only drawing 3 here, but in practice, we have over 100,000 different realities. So reality number 1 is things get wetter, the species likes it. Reality number 2, things get wetter, after very soon the species doesn't like it. Reality number 3, things kind of attenuate very gradually. And our realities in the models that we're using aren't just restricted to how species respond to the environment. They also include dispersal distance, carrying capacity, rates of population growth, and things like that. But just in a simplistic kind of way to illustrate how we're approaching this, I'm just going to use these 3 alternative realities and how the species responds to climate. I'm going to represent these realities through 3 different models, or basically I'm going to use an archer icon. And I'm going to construct a model based on that would basically recreate each of these curves. Then I'm going to ask each of these models to perform a task, and that task is to recreate or predict present day distributions. And that's honestly not a really hard task because after all, I'm giving each of these models data from the present. So they really ought to be able to recreate the same thing that they're actually seeing. Now, I would have better faith in each of these models, or more faith in one of these models, if it were also able to recreate species past distributions. And by past in this case, I'm talking about distributions going all the way back to 21,000 years ago. Unfortunately, we don't have good estimates of species past distributions. Indeed, that's kind of the aim of this whole exercise, to recreate the past distributions. So in the place of having these models kind of shoot the target of recreating a past distribution, we ask them to do something which we hope is equally informative and just as hard, if not harder, and that is to recreate present-day patterns of observed genetic diversity. And this includes over 220 different statistics. For the rest of my talk, I'm going to name all of them. No, of course not. This includes heterozygosity, pairwise FST values, number of private alleles, so on and so forth. The idea being that once we challenge the models to be able to recreate this observed pattern of genetic diversity, some of them are just going to fail. So the arrows are going to fly off into space, but some of them are going to be able to recreate these patterns more than others. And by inference, then we can infer that in this case, in this cartoon example, that model 3 is the more accurate one compared to model 1 and 2. So what I just described is known as approximate Bayesian computation, or ABC. We applied ABC to this species as a case example. This is, again, green ash. I've already shown you some of the aspects of green ash. In particular, this is how we do it, step by step. So step 0 is optional. First, we construct what we call a suitability surface. And the suitability surface is either derived from applying a species distribution model, which is what I'm showing you maps of right here, or a pollen density model, which uses paleopollen or fossil pollen to recreate, in a sense, this the suitability of the surface. And that suitability surface we interpret to be indicative of the species carrying capacity across the landscape. Then, on top of that suitability surface, we simulate demography, and we do that by beginning at one of 3 or all 3 of purported glacial refugia. And I'm not showing you them here, but we have one here in the southeastern portion of the species current range, southwestern. We have one in the southeastern portion, and then we have one that's closer to where the edge of the glacier was in the past, because that's purported to be one explanation for how species were able to keep up with glaciers as they melted. So we simulate the demography across 21,000 years. And then on top of that demography, we simulate, going backwards in time, a coalescent simulation to recreate the genetic patterns that we see today. Note, we're not giving the model any genetic data whatsoever. We're just letting it kind of freely play with the genetics, and then we'll be choosing amongst those models the one that best recreates the genetics. And I don't have a great graphic for genetics, but I'm sure you've all seen what DNA looks like. We repeat this about 100,000 times using different parameters, using different conceptualizations, how the species respond to the environment, different dispersal rates, so on and so forth. And then finally, from those 100,000 models, we choose the top 0.1% that best recreates those patterns of genetic diversity. And we assume that that 0.1% of models encapsulate some sense of true responses to the environment, true dispersal rates, and things like that. So in a nutshell, this is what we did. In a more concrete nutshell, we have a set of models. We have a pollen-only model based on just fossil pollen. And then we have a pollen plus genetics model when you incorporate the genetic simulations into the exact same models. We have a species distribution model based on occurrence data. Then we have a species distribution model based on occurrence data plus the genetics. And then finally, we have I'm going to call it a naive genetics model where there's no suitability surface, or put it this way, we assume that every single portion of the landscape, so long as it's not underwater or under ice, is perfectly suitable to the species. And I'm showing you only like 5 possibilities here, but like I said, each of these is run 100,000 times. So it really represents this huge ensemble of different models. There's a lot of results from this research, but I really have time to get into one of those, and that is how we estimate what we call biotic velocity. That is the rate of species range shifts. And again, to cut it short, I'm just going to focus on the results from the pollen-only model and from the pollen plus genetics model. And here is that plot. So along the x-axis here, we have time before present in kilo years before present. So we're going from 21,000 years ago to the present. And along the y-axis, we have the rate at which the centroid of the species range, the center of the species range, is moving in meters per year. When we run the model only using pollen, not using any genetic data to kind of construct constrain which models we select, we find that, first of all, there's extremely wide variation and uncertainty in the actual rate of species movement across the landscape. It goes up to about 800 meters per year and somewhere down to 0, depending on the time period. So this is commensurate with other people's estimates of range shift based on pollen. However, when we select amongst those pollen models, those that best recreate observed patterns of genetic diversity, you can see our uncertainty dramatically decreases. We have much more precise estimates of the species range shift across time. Now, as you go into the past, we have more and more uncertainty. That makes more sense, I think, because we have less and less pollen data as you go back in the past. But you can see we dramatically reduce the uncertainty in the estimates of the species rates of response to climate change and retreat of the glaciers. And this pattern generally mimics what we would expect based on periods of dramatic climate change in the past, Younger Dryas and Bowling Alleride, when things went climatically, in a sense, very haywire. I would expect higher rates of movement during those periods, and that's exactly what we see. So, end all and be all lesson here is that when we incorporate other kinds of data into what I would call extended species distribution models, that allows us, hopefully, to achieve a higher degree of uncertainty, not only in this case, estimates of species responses to climate change across the landscape, but also the responses to the actual environment. So going back to this plot I showed you before, as you can see in present day climate space, the species niche is truncated. And yet we know the species can exist, or at least the genus can exist, in climates that no longer currently are available in the landscape. So what I'm showing you here is exactly the same slide or the same graph as this one, except that this is pollen deposits. So the gray dots here represent current pollen for the genus Ash, and the open circles, I'm sorry, I don't have a better quality picture. This is the one I could grab. The open circles represent past pollen from about 14,000 years ago. Current pollen is limited. It's only found on the left side of this line. And that's the point of truncation I talked about earlier. But paleopollen, we can find for the genus level at least, existed out here in these climates because those climates existed in the past and the species could, or the genus could access them. Now, here is honestly where I'm a little bit disgruntled. What I would love to show you is the species response to climate as we extend the model out into these climates that no longer currently exist. In other words, it's really filling in the Hutchinsonian shortfall for Ash, the genus at least. Unfortunately, I'm about 2 or 3 months out from actually being able to produce that graph. So that'd be an ultimate culmination of this talk if I could show you that response. But ask me in 2 or 3 months and hopefully I'll be able to do that. So in summary, what we learned is that quite a few species currently live in the sense on the edge of available climate space, and they face climates that have existed in the past and might exist in the future, but because we are all in many cases limited to just occurrence data, we don't have information on the responses to those climates. And that poses a severe challenge to anticipating species responses to climate change and to backcasting them. Luckily, however, at least for some species, I think that a promising way forward is by leveraging extended occurrence data, fossil pollen, genetic data, and you might even extend that, say, to isotopes and things like that. By leveraging that kind of data, I think we can at least narrow the realm of possibility and get a much better picture of how species have responded and might respond to climate change. So I don't want to get all romantic on you, but I'll show you this Romanesque picture. You can think of science as wandering into the mist. You know, we're all standing there as scientists getting along and arguing with one another. And our job is to wander into that mist and feel around for a couple months or a couple years or decades. And hopefully after a while, once we understand something, being able to push that mist away. However, somewhere in that mist is a cliff edge, and we can't necessarily see it. Quite often, we have to extend our knowledge past the edge of the cliff. That's called extrapolation. There's nothing inherently wrong with extrapolation. Indeed, that's what a hypothesis is. You're hypothesizing you're going to see something based on current data, but you don't really know yet whether you're going to see that. That's extrapolation. Nothing wrong with that. But it really, really is essential to demarcating the limit of that cliff edge so that we can say that beyond this point, our knowledge ends and our ignorance begins. So hopefully what I've shown you is a method by which we can, in a sense, use a cane or something like that to feel around and pinpoint where the edge of our knowledge is and where our ignorance begins. And then hopefully I've also shown you methods by which we can extend into that fog, whether it be over the cliff or not, and get a better ascertainment of species fundamental environmental tolerances. Thank you very much.
Well, thank you, Dr. Smith, for a really fantastic talk. We do have about 10 minutes, give or take, for questions here. And I'll remind everybody in the audience to please leave your questions and comments for Dr. Smith in the YouTube live chat. To start us out, our first question here is from Richard Zander. And he asks,
I believe we will have some questions here.
Yeah. So the first one here is from Richard Zander, who asks, have you used the software MaxEnt? Can you recommend it to beginners? It is supposed to do much of the distribution analysis you are demonstrating. Can you hear me, Adam?
Oh, now I can. Yes, sorry.
Perfect, no problem. Our first question here is from Richard Zander, who asks, have you used the software MaxEnt? Can you recommend it to beginners? It is supposed to do much of the distribution analysis that you're demonstrating.
Yes, some of the results I showed you in that second half when I was projecting the response of green ash to past climate change, some of those results are based on MaxEnt. I used 3 other different algorithms. So yes, I would recommend it. It's kind of like, it's the most popular algorithm. It tends to be most accurate, not always for every single species. I'd be happy to talk with you, Richard, if you want to get into that. The software I would recommend for running it is called Wallace. I think it's all lowercase. It's run through R. It's a very user-friendly kind of interface, and it walks you through each of the steps and tells you about the assumptions. And then even gives you references for further reading if you want to make particular decisions about what you're doing.
Wonderful, thank you.
All right, thank you, Dr. Smith. That was really really interesting and amazing talk, so thank you. And so I actually have a question of my own. So I'm interested in whether you have applied these like niche modeling to invasive species. So my thinking is that some of the species, like if they're limited in the geographic range and what climate they can experience, maybe like in their invasive range, you're going to experience some of that, and whether that could, I guess, give the model some insights into how well it is predicting from the current data they're having, and then potentially application-wise, would it be able to predict where the invasive species are most likely to colonize next?
That's a good question. I personally haven't worked yet with invasive species, although it's high on my interest list. I'm very curious about how that method I outlined earlier about identifying niche truncation could inform us about whether or not we know something about if a species could invade a certain area. So there's an ongoing debate in the literature about whether invasive species, when they get to a new location, are successful because they, in a sense, shift their niche, usually through evolutionary processes. That analysis is great as far as it goes, but because you want to do a fair comparison between the environments in the native range and the invaded range, those analyses are always restricted to climates that are the same between those 2 different locations. But of course, invaded ranges often have climates that are very different from the home range. And that difference is ignored because there's honestly, I'm not sure exactly how you would analyze it, but I think this niche truncation kind of measurement might enable us to know whether the species has shifted in niche space in those environments in the invaded range that don't occur in the home range. So I think it has some promise. And like I said, I haven't worked with it yet, but I'm eager to do that.
Thank you. Yeah, that's really cool.
Great. Our next question is from, I hope I'm pronouncing your name correctly, Liana Van Zen, who says, amazing talk. I loved the philosophical ending. How did you gather and manage the huge data sets used in this study?
Oh, good question. You know, for the trees, I didn't use occurrence data. I used those range maps. I did go down the road somewhat of using occurrence data for trees, just those trees that I showed you. There are over 11 million records, and I can process that in this laptop that I have right here. It takes, you know, a couple days, but I'm patient. But I decided not to use that occurrence data because it actually turns out to be extremely biased. In this case, the vast majority of records of North American trees we have come from the US, specifically through the Forest Inventory and Analysis Program. So we have tens of thousands of records of trees, and those records literally stop right at the border. And it's inconceivable that the species ranges stop right at the border too. So I'm going a little bit far from your question, but managing that kind of data takes some thinking through of what you actually need, and, you know, discarding the other stuff because it takes up memory if you don't need it in the moment. Looking forward, I'd like to do this for thousands of species, and I have some data sets in mind, and they are in the order of tens or hundreds of millions of records. And at that point, I will probably have to resort to using a cluster of some kind.
Thank you. Liana also follows up. Also, how do you apply a niche truncation study to migratory species, where the species may exist and spend time in climates they could not survive long term?
Oh, interesting. I assume you mean like when the migratory route, that is, they're just kind of passing through. I hadn't thought of that, but that's interesting. I could conceive of doing this three different ways. I'm thinking of birds here since those are known migrators. You could apply a niche truncation analysis to the wintering range, to the summering range, and then to the transitory range, and see which of those are truncated. Trying to think which would be most indicative of something you'd like to know about. I mean, maybe they're all equally interesting. There are ways to, I didn't have a chance to really get into it, but there are ways to manipulate what kind of climate you're pulling out of geographic space to examine whether a species could, within its current range, experience novel climates, and therefore that would tell you something about the species' ability to persist. And there are other ways to examine whether a species would be able to, or basically characterize our ignorance about whether species would be able to expand into new climates outside of its current range. So those are basically opposite sides of the same problem. So I can imagine you could apply that to a migratory species case. And probably in that case, you'd be interested to know whether or not that migratory route remains suitable to the species, at least during the time that it's used, or whether areas that are climatically adjacent to that migratory route become open and have novel climates, and maybe the species could move through them if the original route becomes obstructed for some reason. Thanks for the good question.
All right, thank you. Our next question is here. I apologize in advance if I pronounced her name wrong. Gerardo Camilo. And the question is, can you speculate about the effect that even faster change like urbanization may have on the living on the edge of Hutchinson's niche?
Oh, very good question. I think one way to fill in that Hutchinsonian shortfall and address those precipices of our knowledge is to use what I would call maybe microclimate studies, not saying a city is a microclimate, but in a sense it is. So Gerardo, as you know, cities tend to be warmer than their surrounding regions. In a macroclimate scale, they don't tend to be too much warmer, but in a particular city, particular places can be much warmer. And we can look in those locations and say, oh, well, look, the species tend to persist or not at even warmer locations than on average across the range. And that would help in a sense to kind of extend past that precipice of niche truncation that we can get at a macro scale by using those kind of microclimate studies. So again, a very good question. Thanks.
Thanks, Dr. Smith. We do have multiple more questions here. Unfortunately, we're right at time, so we'll finish up with what will be our last question, but I'll mention you can always go back to YouTube later and look at the additional questions that people have asked. Our last question here is from Jonathan Lossos, who says, brilliant talk, Adam. My question, to what extent can you incorporate microevolutionary change, either across space or through time, into your analyses?
That's great. You know, all these analyses, except for one, in a sense, assume species are generated through spontaneous generation. In other words, they totally ignore the fact that species are related to one another. We are exploring, like I said, phylogenetic, I would call the phylogenetic SDMs, where you not only look at any particular clade, I'm going to call it a clade, so it could be a species or subspecies or larger group that's related, and how that can be used to inform our knowledge of species responses to climate. So I would love to be able to reproduce some of these analyses, like the niche truncation analysis, but if we had the data, and honestly, unfortunately, we don't for most tree species, but if we had the data, do it clade by clade or group by group, genetic group by genetic group. And it's very possible, I expect to see that certain groups, genetic groups of a particular species might be more truncated than others. And in those cases, we might identify populations that, in a sense, sit on the edge of that precipice and target those for conservation, whether that's seed banking or assisted introgression or assisted migration or something like that. So that's where I think this might profitably go.
Great. Well, thank you. Once again, thanks for joining us today, Dr. Smith. You have many, in addition to the questions on YouTube, many kudos coming in from people in the audience. So thank you again for joining us, and thank you to everybody in the audience for tuning in.
Thank you all.
All right, I have ended the stream.